Welcome to week 6. You've just completed the first section as far as lectures go, so you're 1/4 of the way through the lecture portion of this Bio 201. There were 4 chapters in the first section. There's only 3 chapters in this section. We work through the integumentary system, your skeletal system essentially, and that includes joints. So we start off with the integumentary system, which is chapter 5. So the basic components of your integumentary system include the skin, which is the largest body organ. Think about it, one continuous sheet of tissue. One organ makes up this entire system. A lot of complexity to it, but the largest organ. It varies in thickness, and it's approximately 7% of the body weight. We also include accessory organs, and we're going to start by talking about the 3 layers that make up what we call the skin. So we have our epidermis, which is epithelial or outermost. It's kind of what you see, and we'll talk more about that later. We have the dermis, which is essentially the bulk of the skin. It's tough, it's leathery, it's dense connective tissue, and it's vascularized. And then the deepest layer of our skin is subcutaneous tissue, and it's really not technically part of the skin, though it shares in the protective functions, and so we classify it here. We're going to start, I'm going to give you a quick look at the layers, and then we're going to start by talking about the dermis, working from superficial to deep with the 3 layers. Okay, so again, you can see here you have your 3 layers. This is just a quick general look: epidermis, dermis, and hypodermis. Epidermis being the superficial, the visual layer of your skin. The hypodermis, the adipose tissue, being the deepest portion of the subcutaneous tissue. So we're going to start with the epidermis. So some general characteristics of the epidermis, and then we're going to talk about the layers. This is the most superficial portion of the skin, made up of stratified squamous epithelium. We have some specifics that we'll look at when we look at our layers. Your most superficial skin cells are dead, so the ones that are actually super on the surface that you're looking at, those are technically dead skin cells that are filled with keratin. And the epidermis has 4 different cell types that you can see. We have keratinocytes, which are going to produce keratin for protection, and they arise in the stratum basale, and you'll see them when we talk about the stratum granulosum. We have our melanocytes, or where we produce our melanin. That is in the stratum basale as well. It's part of like what makes up 10 to 25% of our epidermis, and you'll see that on another slide. We have our Langerhans, or our dendritic cells, which arise from bone marrow, and they're there to ingest foreign substances, and they are the activators of the immune system. And then we have our Merkel discs, or tactile epithelial cells, which meet at the epidermis-dermis junction and are associated with our nerve endings. So 4 cell types all arise in different layers of the epidermis. The epidermis, again, as I mentioned, is what you see, and it's made up of 5 strata, or layers, and I've listed them here from deep to superficial, but you can also see them pictured superficial to deep. And when we talk about each of them, I'll follow your notes packet order where I go from superficial to deep. So just kind of taking a look at this picture and what you're seeing from the 5 layers that make up this really thin section of our skin. Again, thinking about the skin that you can see. So first up is our very most superficial, literally what you're seeing when you look at the surface of your skin, is the stratum corneum. These are flattened, keratinized, and cornified Cornification is the flattening and drying of cells. And then friction is going to stimulate the increase in the stratum basal so that it keeps forming like callus formation. So I can repeat that. Hold on. Cornification, you ready? Flattening and drying of cells at the corneum. And then the friction of this is going to increase in the stratum basal mitosis, so the production layer, which we'll get to, and keratin production, which is going to cause the forming of a superficial callus. Okay? But typically here, they're anucleate, and they shed regularly. So they're sloughed off. They're part of what helps build up your dust bunnies at home. They're just all sorts of things that you just shed, and they become really, really obvious in dry skin. So I've listed these two on the same slide, and part of that is because your stratum lucidum is an extra layer that is only found in the palms of your hands and the soles of your feet. So it's only found in thicker skin, and it's a translucent band between the corneum and the granulosum. The cells are pretty much identical to the corneum, so they're highly keratinized dead cells that are flattened out, and they're, again, thick skin, so palms and soles, areas of great use and high friction. Your granulosum are granular cells that produce keratin. We've got our keratinization process that begins in the granulosum. So the cells are flattening, and the nuclei and the organelles begin to disappear. There's no cell division going on. There's not a lot of nutrient supply. So once you add keratin accumulation, if you're on the palm and the sole, you'll see that with lucidum, and then the corneum, and then they're sloughed off. Okay. So as we work our way deeper, we have less keratin and more mitotic or active cell division going on. As we travel from the superficial to deep, we now head into our stratum spinosum. These are living cells, but while they're dividing, mitosis is a bit more limited. Here we house our Langerhans cells, so those cells that activate immune response. These have an interesting shape to them as they begin to have keratin, right, as they head to that granulosum and they begin to flatten. So they're kind of a polygon in appearance. And you can really nicely see in this picture the spinosum and their shape and how they flatten and how keratin begins to appear in the granulosum. That's a good picture of the difference between those two layers. Again, they are dividing, but very limited here, because as we move from this layer to the granulosum to the corneum, right, we have the accumulation of keratin and the end of mitosis and the end of the nuclei, and they become enucleate. So this is the last place we see any sort of mitotic activity. And in the deepest of the 5 strata of the epidermis, we land here at the stratum basal. This is our lowest or deepest epidermal layer. This lies closest to the dermis and is attached directly to the dermis. You can see that in that picture. This layer has good nutrient supply. It is highly mitotic, so cell reproduction is going on. They are either cube or columnar in shape. And from the time essentially a cell is born here, it moves from the basal, the deep layer, through to the most superficial corneum in approximately 27 days. So we see kind of that cyclical process, that monthly cycle of birth to shed of our cells. Here we also have our melanocytes, which are producing our melanin, and our Merkel cells, which deal with our sensory nerve endings. And you can have some stem cell activity here with continuous renewal. So your actual skin coloration is a combination of 3 pigments in addition to all the genetic information. But 3 pigments that you see listed here: melanin, keratin, and hemoglobin. The pigment melanin, the color ranges from yellow to reddish brown to black. It's formed by the melanocytes in your stratum basal or your stratum spinosum. Sunlight's going to increase melanin production and essentially tanning, and that to protect cells, the DNA, from UV light. And freckles and moles have to do with local accumulation of melanin. All humans have an equal number of melanocytes. So skin coloring reflects the kind and the amount of melanin made and retained. Right? And that's where we get that color variation. Carotene is a yellowish orange pigment found in corneum cells and also found in the fatty dermis. You can increase with excessive intake of keratin-rich foods, like vitamin A related for normal vision. And then finally, hemoglobin, which is the oxygen-binding pigment of blood. This is where we get our pink tone secondary to oxygenated blood in the dermis. Okay, so yellow to reddish brown to black, melanin, yellowish orange from keratin, and the pink tones from hemoglobin. A little bit of practical application. The actual color or the hue of the skin also has meaning. So we have things like cyanosis, which is a dark blue color. This has to do with the poorly oxygenated hemoglobin and can indicate respiratory or cardiovascular problems. We have erythema, which is the reddening or the blushing. It can happen from embarrassment, fever, inflammation, or allergy. So there's a lot of reasons that we could get the reddening flush of the skin. We have jaundice, which is a yellow. This has to do with excess bilirubin not being removed by the liver. So you typically get the yellowing. You can see that sometimes in the eyes too, but the yellowing that would indicate some sort of potential liver concern. Ecchymosis, also called hematoma, and I put the word rainbows here because it could be red, purple, green, yellow, right? AKA your bruises. You get blood pooling, you get swelling, and the size and shape of these rainbows of colors actually helps with forensics because it's going to indicate the type of trauma, things like blunt force trauma, and the pattern that the blood is pooling. It can give a kind of a direction to some trauma. And then finally, we have pallor or the blanching, which typically happens during emotional stress because the blood's going to divert elsewhere. It's that, quote, white as a sheet. And then you could also have issues like low blood pressure and anemia to go with high emotional stress. But you know, color does make a difference, and so what you're looking at, the color of the skin, not somebody's skin color, but the coloration of the skin, especially during trauma, you can learn a lot just from color. So here's this really pretty picture. Again, you can see how small the epidermis section really is, and in fact, that's marking it not quite as thin as I really think it kind of appears. But as we transition into the dermis is a much thicker portion of our skin as a whole. So let's talk about the dermis next. So the dermis is our, quote, hide. It's a inner layer. It's much thicker than the epidermis. You see an increased presence of collagen and elastic fibers. We have an increase in vascularity, so blood supply. And then we also contain our nerves, our glands, and our hair follicles here in the two different layers of the dermis. So the more superficial of the two layers of the dermis is our papillary layer. We have our dermal papilla, or our peg-like projections in the papillary layer. It connects to the epidermis. It's thin, and again, it's the more superficial of the two. It's made of areolar connective tissue, and we house our Meissner's corpuscles here, which are our touch receptors. Sometimes you see your free nerve endings, which are pain receptors. We do have some sweat pores here, the pore itself, the opening toward the surface of the skin. And we house our fingerprints. Fingerprints are really interesting. Fingerprints are friction ridge patterns that are genetically determined and unique. Kind of cool. Sweat pores open along their crest, so you leave an identifying film of sweat on anything you touch. So the fingerprint itself is essentially an oily residue along these fringe friction ridge patterns, and that's kind of your identifying film, so to speak. So that's really, I think, interesting about fingerprints is you get these ridge patterns, and then you have sweat pores that open along the crest, and you leave your identifying film on the surface. So our deeper, thicker, more flexible layer of the dermis is known as the reticular layer. 80% of the thickness of the dermis is here at the reticular layer. It's coarse and made of dense, irregular connective tissue. We have some sweat and oil glands that arise here, along with deep pressure receptors known as Pacinian corpuscles. And then we have a couple sets of lines here. Our tension lines, or cleavage lines, are a series of collagen that run parallel to the skin surface. They are externally invisible, and they can run longitudinally. These lines matter mostly to surgeons because if they cut here, they'll actually get less gape with healing when they cut along a tension line, so it heals better. You have flexure lines, which are dermal folds at or near joints. These things like deep creases on the palms, the wrists, the fingers, the soles, the toes. They're just deep folds in what you see. And then we have linea albicans, which are known as stretch marks. This is kind of a tearing of the dermis due to overstretching, often seen during pregnancy, and it leaves this silvery white scars, and you get increased collagen repaired dermis there. So those are some interesting things that you get to see at the dermal layer. And our deepest final layer of the integument is the hypodermis, our subcutaneous tissue. It's going to bind the dermis to underlying tissue, but loosely for sliding purposes. It's primarily made of connective tissue, adipose tissue, and vasculature. And it's a shock absorber and an insulator. And I left this can vary how on here. Well, it's going to vary with body region. Think about us, and think about more native Arizonan and native Eskimo, and the thickness of the hypodermis may vary. It's also potentially going to vary by gender, by age, by nutritional state. So all sorts of things can play a role in the appearance, the thickness, the presence of your hypodermis. There are actually a large number of roles that the integument plays, and you can see 6 general functions of the skin listed here. First up, our most valuable organ system when it comes to protection. We have 3 types of barriers. The first is a chemical barrier. So we have our skin secretions, things like defensin and similar to kind of like an antibiotic. We have melanin, so we also can retard bacteria multiplication. It has a low pH, so we're kind of that protection part there. We have a physical barrier. The actual physical skin is a barrier itself. It's water resistant and water protectant, so it's preventing loss and entry of certain things, so only certain things can penetrate. One of the things that can penetrate our skin that has become more prominent and quite dangerous is fentanyl. Fentanyl can be absorbed through the skin just at touch. That's why it's such a big deal as, you know, kind of the drug awareness piece. And then we also have a biological portion to protection. We have dendritic cells in the epidermis, kind of our immune patrol system. And we have macrophages in the dermis, and they're part of our second line of immune defense. Okay, so kind of dealing with some of those It's like biological warfare. Our skin's kind of protecting us. We also have sensory reception, kind of the part of the nervous system dealing with exteroreceptors, because it's going to respond to stimulus from outside of the body. I've listed here 4 different sensory reception parts, like Meissner's corpuscles, which are tactile, your feel, your Merkel discs, free nerve endings, those are mechanoreceptors. Pacinian receptors feels the deep pressure, and some other bare nerve endings for pain and temperature. And we've kind of already mentioned where those are as we were talking about the layers. So hopefully you picked up on those all playing a role in what our integument does. We deal with regulation of body fluid. We deal with temperature regulation, talking about vasoconstriction versus vasodilation. Vasodilation has to do with radiant heat loss for cooling purposes. So you're going to get that flush of blood rush. We get evaporation of perspiration, so for cooling. And then we get retention of heat by vasoconstriction to help warm. Okay? So vasoconstriction is the shrinking of the vessels or lessening of the blood flow toward the surface through those vessels that run through the skin. Vasodilation is the increase of blood flow. So vasodilation equals cooling, vasoconstriction equals warming. We have cutaneous absorption, which I kind of already mentioned when I was talking about the physical barrier and fentanyl. So many things can be absorbed through the skin. We also deal with UV light, oxygen and carbon dioxide, pesticides. So the skin works for absorption purposes. And then we have a number of things that are synthesized or made by the skin. We've already talked a little bit about melatonin. We've talked about keratin. So melanin is a pigment, keratin for the process of keratinization or hardening. We have vitamin D from modified cholesterol molecules playing a role in calcium metabolism. And then we have an enzyme, collagenase, that aids in the natural turnover of collagen. So that's going to help, obviously, keep collagen in that steady, sturdy presence. On to some accessories. We'll talk about hair and nails and glands. So first up, hair. We have both the function of protection and distinguishment. In the animal kingdom, hair deals with warmth. Humans, we might be able to sense an insect before the bite. We have it on the head for protection, eyelashes to shield the eyes, nose hairs to act as a filter, right? So there's a number of things going on. We have a number of components. You can see the shaft that extends above the epidermis. That's your visible, dead, flexible strands produced by a follicle. We have our hair made of keratinized cells, but they don't flake off. Luckily, our hair would just be falling off all the time. We have the hair root, which is the part that extends just beneath the skin. We have a bulb, which is the enlarged base of the root that sits in the hair follicle. So the hair follicle is the fold down from the epidermal surface, and there are nerve endings there as well, which is why it kind of hurts a little bit if you pull it out. So quick note, hair develops in the epidermis but receives its nourishment from the dermis. Okay, so you can see the shaft above the surface. The root is beneath the skin. It's enlarged at the base for the bulb, and that's its area of cell division, nourished by a hair papilla which sits in the dermis. Okay, so develop in the epidermis, nourished from the dermis. Then we have our color, which is secondary to pigment in the stratum basale. Varying amounts of melanin, right, produce the same kind of effects, dealing with the amount of melanin and therefore the color. But red hair has a unique pigment known as pheomelanin. So that's kind of a unique thing for red hair, naturally occurring red hair. And then, how do we get gray or white? Well, melanin starts to break down, or decrease, essentially. So we have a decrease in melanin, and air bubbles get replaced in the shaft. So it causes it to be gray or white, as in lack of melanin, so a colorless appearance. Interesting. And then your erector pili muscle is a smooth muscle cell bundle. It attaches to the follicle. You can see that attachment on this picture. And it's going to stand the hair up when it's stimulated, aka you get goosebumps and your hair stands on end. And then we have some interesting things that impact hair growth and hair loss. So any number of things can affect growth. Age, okay, got that. Major shocks to your system. So the number of hairs in a resting phase, the growth cycle, vellus versus terminal. So if you have soft light hair versus the head or axillary and pubic due to androgens and testosterone. And then we have this thing called hair loss, which in addition to age or a return to vellus hair, which is that soft hair, could be impacted by dietary deficiency, low thyroid, and drug use. So growth and loss kind of have a number of factors playing a role in the present. But I think still the neatest part of my understanding of the hair has to do with the decrease in melanin and the introduction of an air bubble that makes my hair gray or white. And since I'm seeing quite a bit of that all the time, I find it really interesting. Scale is a compressed stratum corneum that's going to protect the end of the digits. It's like a clear protective covering. It does correspond to claws or hooves in the animal kingdom as a general rule, and it contains hard keratin. So you have a free edge, which is what you cut when you cut your nails. You have a body, or the plate, which is pink. You have the lunula, which is the white part, and your cuticle that sits around the edge there. So not much more I'm going to share with you on that. Just know that it's compressed stratum corneum, and then you've got your free edge, your cut part, pink body, white lunula, and your cuticle. This is a busy slide. We've got a lot of things going on when we talk about glands. Your glands are located in the dermis, and in this particular case, they're exocrine glands, meaning they secrete to the external body. For the most part, they're going to be seeing the results of their work come to the surface. So we have three types. We have sebaceous, which are found all places except thick skin. So anywhere that you have your stratum lucidum, you won't see a sebaceous gland. It's an oil gland, attaches to the hair follicle. It secretes a substance known as sebum, make sure you know that, onto the hair follicle. And sebum is for lubrication and waterproofing. It also prevents brittle hair, and it is bacteria killing. So it's a pretty important substance, sebaceous gland producing that sebum, which is really important. We have our sudoriferous glands, which are regulated by the autonomic nervous system, which makes them involuntary. These are sweat glands. We use them for waste elimination and evaporative cooling. We have 2.5 million of them, believe it or not. And we have two types of sudoriferous glands, right? And we're dealing with sweat here, so we're dealing with water, salt, urea, uric acid, with that kind of substance. We have two types. First are apocrine. Apocrine do not start functioning until after puberty. There's about 2,000 of them. Their true function is truly unknown, but it's linked in the animal kingdom to sex sense glands because they increase in activity during foreplay, they enlarge and recede with the menstrual cycle, and they may kind of act as a pheromone. They're pretty deep in the dermis, and you're talking about an odoriferous secretion, which is why it talks about it maybe acting as a pheromone or a sex sense gland. And their location are really kind of axilla and pubic regions. So it all kind of makes sense. Though again, in humans, the true function is not quite 100% known. And again, they don't start functioning until after puberty. Your eccrine glands form prior to birth and are significantly more numerous. It helps with cooling by secreting through pores in the skin. You do have some of these: forehead, palms, soles, the back. The secretory part in the dermis, and the duct goes through the epidermis to the surface. So the actual gland itself and what's secreting, the sweat is in the dermis, and then the duct travels through the epidermis up to the surface. And then finally, we have our modified apocrine gland here, found specifically in the lining of the external ear canal, to secrete a substance known as cerumen or earwax to moisten the tympanic membrane. And there's lots of other things going on, and I left that listed there because it's not one we spend a lot of time talking about. You're going to get asked about things. It's going to be location and identification of sebaceous glands, pseudoryphorous glands, and then just kind of knowing the difference between an apocrine and an eccrine sweat gland. So with all the functions of the skin, we do have some things that go sideways with our skin, and one of those is skin cancer, and it is the most common cancer with UV radiation as the #1 risk factor. They say something like 1 in 5 will get some form of skin cancer. That doesn't say what level or what level of severity. It just says 1 in 5 will have some form of skin cancer. 80% of those-ish will probably be basal cell carcinoma. It is the least malignant and the most common and most often seen in your sun-exposed areas. It's pretty slow growing. They say that you've got full cure at about 99%. So that's your basal cell carcinoma. So that's your best chance to take care of it. Our second most common type of skin cancer is your squamous cell carcinoma. This is common on the head and the hands. You get the keratinocytes of the spinosum with rapid growth. And you can see the two pictures, they do look a little different. And caught early, this also has pretty good chance for a complete cure. Then you have your melanomas, and your melanomas are most dangerous, and they're highly metastatic, or they are resistant to chemotherapy. They are just an aggressive form of skin cancer. Only about 1% of the population get this, but it leads in death as far as your skin cancer goes. The incidence of this is increasing, and typically if you have existing moles, those are things you should get checked. And that's just kind of typically the rule of thumb, especially living here in Arizona, is to go get your skin checked. And I'm a do as I say, not as I do, so don't ask me when the last time I got checked, especially because I coach swimming and I should be going all the time. So yes, I'll get right on that. Now I have burns listed as a skin imbalance because there is a lot that is going on when you're dealing with burns, and we have a couple things to talk about. The first thing I'm going to talk about is the rule of 9s, and you can see the picture at the bottom there. The rule of 9s has to do with 11 areas, each comprising of 9% of the body, and then the last 1% is for the genitals. Your dealing with burns has to do with tissue damage via intense heat, electricity, radiation, and chemicals that are killing cells, and they're classified by severity. Your first degree burn is a burn to the epidermis. You get some redness, some swelling, some pain. Most of your basic light sunburns are first degree burns. Second degree burns involve the epidermis and possibly part of the upper dermis. This is a first degree burn plus blisters that take 3 to 4 weeks to heal. With second degree burns, we start to think about the threat to life, because it's really starting to think about more of body fluid loss. We get a decrease in protein and electrolytes, which equals dehydration, which equals renal failure and shock. So as we move from a partial thickness or second-degree burn to a third-degree burn, it becomes quite a dangerous situation. Third-degree burns are full thickness burns, meaning we've involved all of the skin. You might see a gray-white, cherry, or black skin with little to no swelling or edema. The nerve endings are destroyed, so typically that area is not initially painful, and oftentimes grafting is part of the treatment, and I'll talk about that again in just a minute. So even if you have only second-degree burns, you could have what's considered a critical burn. If you have more than 25% of the body with second-degree burns, that is still critical. So you get to both portions of the chest and the fronts of both arms, for example. That would be considered critical if it's, again, 25%. So all you need is 9% areas and at second degree with blistering, and we are looking at potentially critical burn. Anything over 10% of the body with a third-degree burn. So just one of those areas of a third-degree burn would be considered critical. And then any third-degree burn to the face, the hands, or the feet is critical as well. The face is because you could be involving respiratory passageways, making that even more threatening to loss of life. So how do they treat this? Well, you're going to get IV nutrients, and you're going to get IV hydration, and you're going to get antibiotics. And then we have two options for dealing with third-degree burns and what I mentioned called skin grafting. They could do skin grafting. That's where they take cuttings from healthy tissue and lay it over the burn. Or we can do synthetic skin. Two different options here. You get silicone epidermis with spongy overlay, and while you culture the dermis, epidermis is going to grow, dermal tissue then will absorb it. It's an artificial application. The other really neat thing that can go on is the growing of your own. So they take a series of skin cells from your healthy skin, and they grow them into sheets, which is really neat. So it'll go through rapid mitosis, and then they overlay your own lab-grown sheets of your tissue from your cells with the burn area. And it's kind of the same process as synthetic skin. They're going to overlay it, and as it develops and grows, the dermal tissue will absorb it and push layers of epidermis to the surface, and you'll get healthy tissue return. So those are options. The last two slides on here are just other views, other looks at diagrams of the skin. They have some different things shown on them. I would start to become very familiar with how these are laid out and what's located where. Become familiar with all the structures listed on this slide. This one has some of your layers of the epidermis labeled, and just your general layers of the skin, epidermis, dermis, and then it's labeled a subcutaneous layer on here, but just know that it's the hypodermis. And on this last and final slide, again, you can see the three general layers. It labels some more glands, ducts. Your erector pili is labeled. You can see the nerve fibers, the hair follicle itself. So again, it's just about getting to know all these structures and where they fall, what layer of the dermis, know those layers of the epidermis. The quiz asks you lots of things about the layers and the synthesis, what's synthesized, your glands, putting your strata in order, right, the pigments. So the quiz is really, it's been covered in here, so take your time when you go to take the quiz. And as always, don't forget, you can email anytime, you can set up meetings, you can email me your questions, you can post on the discussion board, anything you need to help you be successful. Hopefully the first portion of this has gone reasonably well. If not, we can always talk, set up a meeting, and we'll do our best to move along together.